Hi everyone, welcome back to my video. So glad you're here. So we're going to be talking about DNA replication today. When your cell is ready to go through cell division, mitosis, everything is duplicating, and so is your DNA. So DNA needs to replicate. So here we have DNA helicase, which is an enzyme that will basically cleave the hydrogen bonds and separate those two strands to produce another strand inside. So we're going to be talking about DNA polymerase later on, which helps make your strands, your new strands. So as DNA helicase is removing those hydrogen bonds and it's moving left and as well right, you have your DNA, which is twisting and turning as well. So you can think of it as a rubber band that if you tighten it up really hard and you're twisting it and then you let go of one side, it twists back to be straight like this. So I hope that rubber band analogy kind of helped. And because of this twisting and turning that happens in the DNA molecules, it creates a lot of tension, and we're going to be talking about another enzyme that will help fix that problem. And as DNA helicase is moving left and right, more DNA is being exposed. So now we're going to move on and talk about more specifically about how DNA replication process actually happens. All right, so here we have a more simplistic version of it. So I'm going to zoom in right here on the enzymes. First, I'm going to be talking about the enzymes, and then we'll go on to the animation. So we have our DNA polymerase, which builds the DNA strands itself. It has two sites, the editing site, and then it has the synthesizing site. So DNA polymerase can actually proofread its own work as it's building the strand. So that's kind of cool. We have a RNA primase that allows DNA polymerase it basically tells DNA polymerase where to start replication. And then DNA ligase will basically glue two strands together. We're going to be talking about that later. And then we have DNA topoisomerase, which relieves the tension that is produced inside of the DNA strand, because as DNA helicase is going, it's actually uncoiling the DNA. It's the DNA helix is basically a helical form, right? So as it's uncoiling all those two strands, as it's separating the strands, it actually creates a lot of tension. It's like a rubber band, basically. When you twist it so many times, it gets really tense. So DNA topoisomerase basically cuts one of those strands off and glues it back together again. That helps relieve the tension that's built up in the rest of the DNA strand. And I forgot to mention, there's this sliding clamp on the DNA polymerase that basically allows DNA polymerase to stay onto the DNA strand, and it helps it slide along so that it doesn't, fall off. We're gonna go to our animation now. I've drawn here a little sugar molecule just to show you which carbon is which. So we're going to be starting at the 5' right over here, and then the 3' is here, and then 3' here, and then 5' here. So usually this helicase molecule, DNA helicase, it will start at the replication origin, and it moves in two directions. One way is this left, and then the other way is towards the right. But we're mostly going to be just focusing on half the side, just to make it even more simpler. So we have DNA polymerase going from 5' to 3'. And then the other version would be going from 3' to 5'. So one thing to note about DNA polymerase is that it starts at 3'. So it'll start right here. I'll just draw a little molecule. And it will build a continuous strand from 5' to 3'. The new strand will be the new strand will start at the 5' and move to, and it will build towards the 3' end. So the DNA poly will basically recognize this 3' spot. Won't recognize it, sorry. I forgot to mention another thing, that RNA primase will help DNA polymerase start at that site. So it will basically lay down an RNA primer which has U's, so uracils, instead of T's. That's an RNA primer. So DNA polymerase comes here, starts synthesizing from 5' to 3'. So that's your new strand built. However, over here, we also have DNA polymerase, but it has to build another strand, which is going to be 3' to 5'. But like we said, DNA polymerase can only build from 5' to 3'. So how can it do this? That's the tricky part. This molecule called RNA primase will lay down, I'm going to zoom in further, will lay down a RNA primer right about here. Your DNA polymerase will go ahead and start right here at one of the sugar molecules, which is 3'. It'll start right here and it will build a DNA strand consisting of DNA nucleotides going towards the primer. So RNA primer will be set about right here. You have your molecule coming in, your DNA polymerase, and it builds it from this way to this way. It starts at the 3' end, goes towards the 5' end, building from 5' to 3'. My bad about that. And RNA primase will come in again and set another primer. And you have DNA polymerase that ended right about here. It will come in, go this way back to the RNA primer that was set, build another set of DNA nucleotides, and continue. So as DNA helicase, this molecule right here, as it's opening up more, as it's exposing more DNA, RNA primase will continue to set its RNA primers, and DNA polymerase will continue to create new fragments. So these fragments right here, they're called, this little fragment that's been created, that's an Ozaki fragment, because it consists of RNA primers and then you have DNA nucleotides as well. It's very important that DNA polymerase goes from 5' to 3', because that's what it's conditioned to do. It can't build from 3' to 5'. That's not in its coding. So that's one way that it does it. Sorry, that's the only way it does it. It's a little bit of a tricky concept to understand. So now we have RNA primers inside of our DNA. We cannot have uracil in our DNA. We all know that. We can only have thymines. So one way that our body basically removes those RNA primers is through 3 steps. So I've listed it down here. Whoops. So one way that it fixes this is nucleases will come in. So the first step, nucleases will come in and eat up those RNA primers. So now your RNA primers are gone. So now you have these gaps in between. You will then have a repair polymerase come in and add in your DNA nucleotides consisting of thymine. Now, as you can see, there's gaps inside of the DNA. The 2 strands need to connect together. So we have DNA ligase, which was our enzyme that we initially talked about. That will come in and help these two strands, so the white strand and the red strand that we used, to bind together. So it uses ATP hydrolysis to do that. So now you have 0 gaps inside your DNA, and it's a continuous strand. So there are two other terms that I also want you to know. One is the not the template strand, the leading strand. Sorry about the bad handwriting. And the lagging strand. So lagging would basically be where you're creating your Okazaki fragments. So this is going to be your lagging strand. And then your leading strand is going to be your continuous from 5' to 3'. So the DNA polymerase is able to continuously build your new strand from the 5' to 3' end. So this is your leading strand. One thing that I want you to all note is that I totally forgot to mention the DNA topoisomerase. That molecule will basically help you, so it'll make the next, like I explained earlier, it'll make the next inside your DNA strand. So it'll cut it right here, and well, not there exactly, but it'll cut it after the DNA helicase to relieve that tension. Another molecule that also helps the DNA fragments, is SSBP. So that's called single-stranded binding proteins. Your hydrogen bonds within your nucleotides are really, really strong, that they want to come back together again. So these two strands that we have right here, they want to recoil. They want to come back together and create those hydrogen bonds, because hydrogen bonds are really strong, right? So these single-stranded binding proteins will come in and bind to your two strands and basically stop that from happening. They'll stop the complementary base pairing from happening, because we need to keep this whole site open in order for DNA polymerase to come in and create those new DNA strands. So these SSBPs are very important in doing that. They're called single-strand binding proteins. So they only bind to one of the strands. Only one strand. They don't bind like over here to both strands. That's what I mean. So I think that's about it for this video. If you guys have any more questions, I know it was a little bit confusing process, but please write down in the comments if you found anything, confusing, and I'll reply back. Thank you.